Okay, that is 4:30. Let's see how this goes. Okay, stream health looks like it's in excellent condition. If anybody would like to say whether they can hear me or not, I've got it all set up, but this is the first time I'm doing this. So, you know, to quote that dog meme, I have no idea what I'm doing. For anybody who doesn't know me, my name is Ben Ward. I'm a maintainer and developer of a not insignificant amount of the BioJulia ecosystem, and I thought I would do some streams showing you some of the some of the tools that are in development in BioJulia. My obsession recently has been developing BioJulia to the point that you can do your genome assembly in Julia and analyze your GenomeGraphs. So I'll begin with, I'm quite quiet. Okay, let me adjust this then. How's that? How's that? Is that better? I've raised it a bit. This is in OBS, so I've raised the volume. I don't know if I have to raise it anywhere else on my Mac settings or something. Oh, Cam says it's better. Okay, let me see if there's anything in my system settings that are holding me back in particular. Yes, I can raise the input volume a bit more on the system settings as well. Okay, brilliant. Okay, so where was I? Okay, genome assembly. So for anybody who is not particularly familiar with this problem, so you're watching just because you're interested in Julia, genome assembly is the problem of reconstructing a model of the genome of an individual or an organism you're sequencing from smaller segments of sequencing data that are called sequencing reads. So I've got some slides on the screen here from the de novo assembly training we do at our workplace at the Institute. And it's visualized here. As you can see, the original DNA molecule is pictured as a red line at the top there. And if we had a perfect world, you would have some kind of machine that you could, you know, feed one end of that DNA molecule into the sequencer, and it would spit out one string that covers the entire genome and has the letters of the genome in order, so you would get the complete genome sequence out of it. Now, we don't live in that world, so actually what we have is a situation visualized just underneath. Actually what you have is you have a DNA sample extracted from an organism, and because DNA is actually quite fragile, what you have are a load of fragments of DNA, and then the sequencing machines we have actually only sequence so far in from either end of those fragments. And so you can think of it as a sampling problem, right? You have an original DNA sequence, and then what the sequencing machine gives you are these so-called reads, and it's like every read is like a small sample of the complete picture. In the same way that an ecologist wanting to figure out, the composition of animals in a woodland somewhere would have to. You can't do a sensor of the whole woodland. You'd have to put down small quadrats and record what's in those quadrats. So it's a sampling problem. And with assembly, what we're trying to do is take those samples and put them together into longer sequences. So that's the genome assembly problem. There are 3 hallmarks of a correct assembly, a way of 3 ways of distinguishing whether your assembly is good. So it has to have the right motifs. So the motifs in this case are k-mers. We'll see how you generate k-mers in a little while. But basically, if you imagine the genome has a massive string, yes, so Adita Puranik says, and assume the fragments are corrupted once in a while. Correct, they are. And depending on the genome sequencing technology, they are corrupted in different ways, right? So the machines have errors, and and the errors occur in different ways. So there are machines that do paired end, so Illumina machines, for example, do paired end sequencing data where they sequence in from either side of a fragment of DNA, and they have an incredible accuracy, actually. Like it's 99.9% accurate or something like that, which is why actually in our group we recommend paired end reads to build your first assembly graph, because the base content, you need a good starting base content. But what happens is these machines run on a chemistry, right? So basically the DNA gets sequenced by a chemical reaction which copies the DNA, and as the reactions, there's like flashes of fluorescent light that the machine picks up. And so the longer that reaction goes on, the more the reagents are used up, and the more the chemical reactions, because they're all happening in parallel on this platform, the reactions they start out at the same. This one cluster is sequencing, there's one reaction that's sequencing the first base, then the second base, and so on. And that's all happening in parallel. But as time goes on, they all get desynced from one another as well. So basically, the longer the chemical reaction goes on and the process continues, the probability of the strings getting corrupted, if you like, increases. And it gets worse than that, because there are certain motifs as well that the sequencing machine, like particular motifs that will mess with the mechanisms by which these various sequencing technologies work, and will cause errors to happen much more frequently. And there are other considerations as well. So you have, that's just at the kind of machine level, right? But then you have to have software that interprets the signals coming from those machines and turns it into the base calls and all this kind of thing. And those software could have bugs in them or not be able to tell one base from another in different circumstances. So there are sort of levels where errors might get introduced in this process. So, but going back to the definition of a correct assembly, the right motifs in this instance is, if you imagine in Julia, your genome is a great big string. The motifs are all the substrings, all the overlapping substrings. So the genome has to have the correct motifs first. If there's a motif in there that is wrong because, you know, one of these fragments, the read of one of these fragments is corrupted, then, you know, it's not correct. The motifs have to appear the correct number of times in the genome. So, you know, it's always important to remember that a genome is not a random string, actually. It has structure, it has function, and there are motifs that appear more than once in that string. So, you know, in that genome string, if you like, you know, there are common sequences, common motifs. So there'll be like promoter sequences, which appear sort of upstream of a gene sequence, a gene motif, and it's their job to activate the gene and stuff when the genome's doing its biological function. So they can appear multiple times in a genome. You might have certain protein domains that are coded. Genomes have interesting evolutionary histories, so they might have. There might have been an event during evolutionary history, where, you know, a segment of the genome got duplicated and copied. So there are all these things. It's not a random string, not purely random, and you have organisms have different levels of ploidy, right? So something like an E. coli bacteria has one copy of its genome, but we humans, we inherit a copy from our two parents. So, you know, most motifs in a human actually appear, or a lot of motifs in a human appear twice because they've inherited two copies of the genome. So in effect, the human has two strings for its genome, as opposed to one. So yes, the right motifs, the correct number of times, and then in the correct order and position, because you need to put the motifs in the correct order and position to get the original genome. So you'll notice that none of those three things are actually quantified or assessed by the various stats that are reported a lot in genome papers like N50 and this kind of thing, which only tell you sort of how large the various strings that a genome assembler spat out are. And everybody obviously wants a more contiguous genome, and they want more of the motifs put into longer and longer strings, because it's useful for, you know, knowing about structure and function and this kind of thing. But you could have an arbitrarily contiguous long genome sequence, but if it's not correct, then you can make the argument that it's not doing you much good, you know? I mean, it's a bit like, you know, saying, you know, when you're coding things, if you're coding things for speed, you can make it arbitrarily fast if you don't care what the answer is. So there's that kind of trade-off to be made. So one of the algorithms to put these fragments back together into the original genome sequence is called overlap layout consensus. And the idea is you have all your reads, and you're basically trying to figure out which ones overlap, and then you're trying to put them in the order, and then from that order you create like a consensus. So you can see there's three stages: overlap, layout, and then the consensus at the end. That's not normally used for de novo assemblers that start with short reads. It's normally more kind of long read assemblers and things like that. But as a general rule, I'm going to recommend that people who are doing genome assembly should generally start from short reads because they're more accurate. There are exceptions, so you know, there are always exceptions, and it depends on your problem and your data. But in general, you want to start with short reads generated from one of these short read sequences that I've already mentioned. And then if, when you create your assembly, there are parts of the assembly you can't resolve, you can't put in order, then go to the long reads, to technologies that give you longer reads, and use those long reads to untangle and sort out the harder to assemble parts. And the reason is the long reads, as useful as they are for giving order, they're not accurate base. They're nowhere near as accurate base by base currently. So it's better to have an accurate graph than use the inaccurate long reads to try and sort out an accurate genome assembly that is quite fragmented, and then use inaccurate long reads to sort out the ordering and resolve more of your genome than it is to start out with an inaccurate genome. The method that I'm going to demonstrate today, how to construct your sort of an initial genome assembly in Julia, uses a method called the Bruijn graph method. So that is basically what you do is you have your sequencing reads, you decompose them into shorter motifs, the short overlapping motifs, so your k-mers, and then you construct a de Bruijn graph from those k-mers. And then the true genome is contained in that graph. There will be a walk through the graph that if you did that walk, you would recover the genome, assuming that your sequencing data is good and all that. And we'll talk about what it means to have good sequencing data in a minute when I start to show you, when I start to walk through some Julia code with you. So there's a couple of slides here that just visualize the process of assembling a de Bruijn graph, right? So you have two sequencing reads here, seq1 and seq2, and what you do is you take, this is using a k size of 3, so the overlapping motifs being used to create the graph are 3 base pairs each. You can see we go through the read, and as we go through the read, every distinct motif is being added as a new node to the graph. Okay, and when we get to the second read, we add some more nodes to the graph, but then we hit a motif that we've already seen, this TTC, because we already saw that motif in the first read, and now instead of adding a new node, we're just updating the count of that node. And then it progresses to the end, and you can see the 3 nodes in the middle now have a count of 2 instead of 1. And if you did a walk through that graph, you would recover the sequence, the full sequence. And let's say you added a third read, and let's say it had a base in it which was different. So this could be a sequencing error, perhaps, or it could be a genuine polymorphism, because the genetic content that you inherit from one parent will have a few unique differences, some polymorphism, to the genetic information that you receive from the other parent. And so it could be either this, so here you can see this sequence 3 has a G in the middle, whereas up until now we've observed that there's actually a C in the middle. Now that could be because a sequencer made a mistake, or indeed this kind of heterozygosity, call it this, a genuine difference in something like a diploid organism, like a human, where they've inherited 2 different sequences that have a genuine difference. So how would that look in the graph if you added this read to the graph now? Well, you would start again going through your k-mers, updating the counts as you go. So you see the nodes on the left now have counts of 2. And then once you start hitting motifs that incorporate that different letter, it creates a branch in the graph, and it continues until you hit the motifs that you've already seen again, and so it creates like a bubble. So genetic differences and errors between, so genetic differences between haplotypes, if you're working with organisms that have, a greater ploidy than 1, and sequencing errors can create these bubbles. And you see and now there are 2 ways of walking through the graph and recovering sequence. You can go sort of along the top, or you can go along the bottom and then rejoin. and that gives you two different sequences. Okay. And sequencing graphs can get quite complicated depending on how complicated the genome and how complicated the organism is. So you can see there's this fairly nice path going along, and then it suddenly gets to this knot in the middle, which is just, how do you find your way through there? So there are common structures, right? So there's a polymorphism, which causes bubbles, like you've just seen, how a polymorphism might affect the construction of the graph, forming these bubbles. You can get erroneous base calls. These cause a structure that we call tips. Now, sometimes errant base calls also cause the bubbles that polymorphism cause, and there is a way of distinguishing between the two. The reason why they cause tips is that, if you remember, I said a lot of sequencing technologies, the chance of observing an error increases the longer the reaction goes on. That has the offset of most errors occur at the end of, if you have that kind of read, most of those errors occur at the end of the read. And so basically, as you're constructing or adding nodes to the graph from a read, when you reach near the end of that read, you start hitting motifs that contain the erroneous bases, and that causes a branching, like you saw in the demonstration where the bubble kind of branched off. But unlike the bubble, you don't rejoin, because you get to the end of the read and it's errors and you have nowhere to go. There's nowhere to rejoin because you've hit the end of the read. So they cause these tips. And then you have repeating elements, which, these kind of things, it's where you have two separate paths through the graph, and they collapse into a single one and then branch out again. These are caused by repetitive content in the genome. So it might be that maybe the first time round you pass through that region going from, one of the top inputs and then through that collapsed region and then out the bottom, or you can come in and out of that region a couple of times through a few different nodes. And again, that's because there are these regions of the genome, these motifs in the genome that are repeated. But because you've broken all the reads down into k-mers, and you don't add, every motif is represented by exactly one node in the graph, it's all collapsed. So the repeated motif exists once in the graph, but to do your walk, to get your original genome out, you have to go through it several times. So that's a repeating element. And so what you normally get at the end of the graph construction process is something like this, and then you basically use a couple of different heuristics to, you might clip the tips off, you might remove some low coverage nodes, remove the bubbles, and then you have a nice neat path, and then that can be, your genome sequence. Okay, so that's all I want to show for these slides and things. Let's actually do some of this Bruijn graph construction in Julia. So allow me to switch to a Julia terminal. Okay. So I'm going to be using a recently developed package called GenomeGraphs. Okay, which is going to be the base framework for a lot of this kind of graph-based manipulation of genome as an analysis of genomes going forward, as we do as more and more tools are developed for BioJulia. And I'm also going to use a couple of other packages. So we're going to do a little bit of visualization. So I want to be using Makey for this. And I also, there's another package which is not included in GenomeGraphs right now, but maybe it will in the future. Chemoanalysis Mackie, and that just has some custom plotting recipes in that I've defined for analyzing chemo spectra. So the first thing we need to do is we need to prepare our sequencing reads. So I have some reads in 2 files from some paired-end sequencing, and we're going to be using some reads from E. coli. So what I'm going to do is they're output by a genome sequencer or a genome sequencing pipeline in these formatted text files called FASTQ files. And because it's paired-end sequencing, there are 2 files, because the first file contains the first read of every pair, and the second file contains the second read from every pair. And what is a pair? A pair is, well, if you think back to the schematic I showed where you had a fragment, these purple fragments, the sequencer generates reads from either end of that fragment. So every pair of reads comes from one DNA fragment. So I'm going to construct a data store for these reads so as we can work with them. So the data store is a container that's backed by disk, and it contains reads. And it's basically so as you can store them and manipulate them as if they were kind of like a vector, you know, a load of sequences in a Julia vector, but actually it's not all stored in RAM because read files can be quite big. So I'm going to use this function read data store. I'm going to pass the first file, so I have them in a folder I've called test here because I've been using these paired-end files to test as I've been working on the package, and then I'm going to include then test E. coli paired-end r1.fastq. Sorry, not r1. Aha, typo. r2. And there are a few other parameters I want to pass to the data store. So I'm going to pass in this. We're going to pass in this string E. coli dash parent, or PE for parent. So this is the name of the file on disk that backs this data store. And now we're going to provide some numeric values. So 250, 301. Now these are the minimum length of a read and the maximum length of a read. So paired-end reads are very, very consistent in length, but they do vary somewhat. And so what this minimum length of 250, as this read data store is created, what's going to happen is it's going to iterate through every read pair, and if one of the 2 reads is shorter than 250 base pairs, it's going to discard that pair. Conversely, for the maximum read length of 301, every read pair it analyzes as it builds this data store. If any of the reads in the pair are longer than 301 base pairs, it will truncate the reads. So if you have reads shorter than the minimum, they're going to get discarded. If you have reads longer, it's going to truncate them. So it's a good idea to choose a pair of values that capture the length of the vast majority of your reads, because if you misspecify that, you could end up throwing away a lot more stuff than you need to. I'm going to put another numeric value here, 0. Now 0 here, this value is the insert size. So again, thinking one fragment of DNA being sequenced from either end. Because the sequencer doesn't cover the full length of the molecule, there's a chunk of DNA in the middle that isn't sequenced. And that's our insert size. I've set it as 0 because right now, for this data set, I actually don't know what it is. And also it's not important for anything we're doing today, anything that I'm showing you today. It's sometimes useful for some algorithms where you start to, once you have your first genome graph constructed, you're starting to untangle the more complicated bits and all that, but we're not going to go into any of those heuristics today. I'm more interested in just showing how you construct your first graph. Okay, and now the final thing is FWRV. Now this is a kind of a mode specifier, if you like. So there are two kinds of reads that can be represented by a paired read data store. You have paired end reads and you have long mate pairs, which aren't used as commonly as they once were because of long read technology. But basically the difference between a paired end read and a long mate pair, a pair of long mate pair reads, is that, again, you have your fragment. With paired end reads, you have the first molecule reads in from one end and the second molecule reads in from the other end. And in your read file, it's the first read is in the forward. You might have to, if you're unfamiliar with DNA, right? So actually, DNA is double-stranded. So if you have your double-stranded chunk of fragment of DNA, read 1 will be on the forward strand, and the second read of the pair will be on the reverse strand. Whereas with long mate pairs, it's like the opposite. So the sequence is at the ends as well, but instead it kind of goes from inside the read out to the end, and like that in the molecule. It's kind of weird how it works, and it works. The reason it does that is because of some steps in the library prep which like change the orientation of how the sequencer sees them, but it doesn't really matter the exact details for now. Basically, FWRV means regular paired end data store, whereas if I specified RV forward, that would mean long mate pairs. So again, much like the 0 value, this is not currently used for a lot. So it's going to tell me that it's building the paired read data store from FASTQ files and that it's writing those reads to the data store. And you can see no reads got truncated, and some read pairs did get discarded because one of the reads was too short, but yeah, that's okay. Can live with that. So we have our reads, and the next step is to decompose them into those short overlapping motifs, those k-mers. So the way you do that, first, what I'm going to do is create a k-mer counter to do that job. Now, k-mer counters are kind of like function objects in this framework at the moment, because there are different kinds. Like k-mer counting is one of those things that is conceptually very, very simple. You're just going through all the k-mers and counting how many there are, right? But because of the volume of data, there are actually like quite a few different strategies and optimize. Like it's a process that you kind of want to optimize depending on your needs. You know, some k-mer count software do it like multi-processed. Some use multiple threads. Some do things in like disk batches and stuff. And it's just because of the volume of data that has to be broken down into these k-mers and get counted. So thankfully, E. coli is quite a trivial data set, and this is a reasonably good MacBook Pro. So I'm going to construct a k-mer counter that basically does everything serially, so there's no multi-threading or anything like that, and it does it completely in memory. There's no batching to disk or any of that cleverness coming on. It's just I can fit the whole problem in my laptop's RAM. So meh. So I just got a question. Do you use GPUs for this graph construction? This algorithm does not. I could imagine that you could do it. The algorithms that are in GenomeGraphs at the moment don't do that. It's all CPU, and it is actually serial right now. There is, I plan on adding some multi-threaded stuff to speed it up. If people know how to construct a graph like this from GPUs, I'd be interested in talking to those people. I don't tend to use GPUs that much day to day though, so it's not something I kind of leaped for when I write any of this stuff, especially when it's like early stages and it's like I'm developing it rapidly, pushing stuff, doing minor updates and stuff every day. I'm just not really used to doing it. I've tried using some of the array fire stuff so it's just like a drop-in replacement, but it's not something I'm familiar with. I'm not sure how I would translate the current algorithm and put it on a GPU because I'm just, I don't have experience doing it. It's something I'd like to learn because I know there's a lot of GPU like extensions and stuff in Julia, like say the array fire and all this kind of thing, that make it easier. And there's like the CUDA packages and all that kind of thing. But it's just something I don't get around to using. It's kind of, I know it's there and I keep saying, I should, get onto that and get into GPU stuff. But it doesn't happen because there's always something else I have to do. So so for now it's CPU. If anybody's interested in having it use the GPU, I'd be interested to see. So I'm going to create back to the Kmer counter. I'm going to create a Kmer counter. I'm going to call it SM, and I construct it by saying serial mem because it's a serial and in-memory Kmer counter. I want to use DNA. I'm going to use Kmers of, so in the schematics it used like these little 3 base pair things. I'm going to use 31 base pairs, and in the BioJulia framework, Kmers are a sequence type, and the length of the Kmer is a parameter of the type. So here you've got DNA mer curly brackets 31, because it allows a lot of stuff to get optimized out when it's part of the type. And it also means you can't mix Kmers of different sizes in the same data structure, which is sometimes a good idea, because the representation of a Kmer is basically like a uint64, and a lot of software where they just generate an array of like uint64s and stuff, but it's like, if you don't have that type to restrict stuff, you might combine things that shouldn't be combined and stuff, and like start comparing 31mers to, I don't know, 27mers and stuff, and it might not be valid to do that. So at the moment it's like quite strict typing. Hasn't been a problem so far. So I'm going to construct a serial mem Kmer counter, and the second thing I'm going to pass to it is this canonical. So this is like a mode again. So when you count, so Kmers, much like other DNA sequences, because it's double-stranded, you have a forward and a reverse orientation. So actually every Kmer in the universe of Kmers can be represented by half as many Kmers, because for one Kmer you also have another Kmer that exists that is like that Kmer on the other strand. So we're going to say canonical, so it like collapses all that down, and you only create a graph of canonical motifs. And in this case, a canonical sequence or a canonical Kmer is just the one that is the lexicographical less Kmer. So in the implementation, in the Julia implementation, it's just a less than. Is this Kmer less than this one? Done. That's it. So create the Kmer counter. You can see from the message it prints out. It's like a function object. So now I can use it to count the k-mers in my reads. So let's call them read k counts. I'm going to do sm brackets ds. So I'm going to use the sm serial mem k-mer counter to count the reads in my ds, in my data store. Let's go. And this will take a few seconds. This is one of the k-mer counting for building your deep Bruijn graph. This is one of the meatiest steps. So it takes it doesn't take more than a minute free cola on this laptop actually, even when I'm doing no parallelization. I'm doing it all in RAM and everything, you know, not doing any kind of fancy optimizations. There will be other k-mer counters in the future as I work on the API. There's already one that actually exists, but it's not kind of exported in the API yet because I revised the API. But basically it's the same, so it's all in RAM, but it's multi-process. So I might do another one which is again all in RAM but is multi-threaded or something. So you can basically pick a mixture strategy for doing the counting. Okay, so as you can see, it's spat out a big array of these k-mer count structs, and it's pretty printed them, so you can see like the k-mer that is A, you know, all the A's occurs 107 times. The one beneath it, A ending in a C, occurs 5 times, and so on. It's gone off the screen, so allow me to just, there we go, otherwise you won't see what. Type next. So the next thing to do is we're going to analyze this k-mer counts. We're going to do something. We're going to build what's called a k-mer frequency spectra, and this is where it helps to get back into thinking of, again, those fragments, and then the sequencer gives you little samples of those fragments. And the idea of sequencing, DNA sequencing, is like a sampling problem where you obtain all these tiny little your data set is tiny little samples of a larger picture. So we're going to construct the k-mer spectra, and that's really easy. And that's just using a function that's called spectra. So I'm going to say read underscore spec is equal to spectra of the read k counts. And that's already done. So this is basically like a frequency histogram, if you like. So you can say, if you wanted to know how many k-mers appear one time, you can do this. You can say read spec, you know, how many k-mers appear once. And there's, well, I have to print it because it stores the values as uint60 as uint. So you know, if you wanted to see how many, you know, k-mers occurred 10 times or no times, or, you know, obviously there are no k-mers appearing no times, but the 0 is there for later. So you have this k-mer. So let's do some visualization then. Let's do some plotting. So I loaded Mackie and k-mer analysis Mackie earlier. So there's a type recipe for this type. And so I hit plot, and allow me to just transition the scene so as I can show you the plot. Okay, so the x-axis is obviously, the x and y axes are obviously a little bit too big, even though there is some functionality in the plotting function to kind of limit the scene, because as you can imagine, the array, the underlying data of the histogram can have a lot of zeros and this kind of thing. So we're gonna, let's do some, you do this is how you do it in Mackie. If you want to customize an image, you do limits. I'm going to construct an f rectangle, let's say 0, 0, 100, 500,000. So what this is going to do is basically limit the x and y, limit how much of the x and y axis you see from like 0 to 100 on the x-axis and 0 to 500,000 on the y-axis. Okay, so we have our limit, and then let's just replot our read spec using these limits equals limits. Okay, so that's a bit better. So looking at this plot, a couple of things should immediately jump to your attention. The first is this, where the x-axis equals 1, you know, the bars shoot off the visible area. Now, so these are all motifs, all the k-mers in the reads that appear once, twice, you know, 3 times. These are the k-mers that are low frequency, and they are probably caused by sequencing error, right? Because if errors are random, whenever you decompose a read that has an error in it, the k-mers that cover that read, because the errors are random, they probably just create these like random k-mers that only appear once or twice in the whole data set. So this is again where you thinking about it as a sampling problem. It's kind of like they're outliers in which you've observed something that isn't actually part of the rest of the distribution. So you can see then, so those are the errors, and the big nice really like neat distribution centered at, you know, x equals 40, 45 maybe. That's your actual distribution of all the motifs that you've observed. So this is like your the good content, if you like, the useful content in your sequencing data. These are all the motifs that have appeared and you've observed enough times to be confident that they probably belong in the genome assembly. And what is nice about this plot is there is a clear distribution, you know, somewhere between x equals 0 and x equals 20. There is a clear separation between these two distributions, this error distribution, or this low frequency distribution, or distribution of low frequency k-mers. It's the, you know, correct way of saying it. And this distribution centered around x equals, you know, between 40 and 45. And you'd be unsurprised, anybody familiar with genome sequencing will be unsurprised to know that when you do a DNA sequencing experiment, there's this parameter called coverage. And again, it's like thinking of it as a sampling problem. You know, if you did an experiment in which your expected coverage is 50x, 50 times, you expect every motif in the genome, everything being equal, every motif in the genome, every genuine motif in the genome, to have been observed 50 times, because it should have been covered 50 times. And so the fact that this distribution occurs sort of, you know, between x equals 40 to x equals 60 isn't surprising. I think this data set was covered at something like 50x. So that's not surprising, and it's good that we see this nice distribution centered roughly where we expect it to be. So when we build our Bruijn graph, we want to include all the k-mers that are represented by this main distribution and exclude all the k-mers, you know, at the low x values. So let's do that. I will shift our screen back to so you can see my terminal. Okay, let's do it. So I'm going to use one of Julia's array comprehensions to do this. Let's create a variable. We're going to call it filter k-mers. So this will be our filtered k-mers. And so actually, before I do that, I'll show you the read k counts. Let's get a k counts. Let's get the first km count. See, it says AAAA occurs 107 times. Well, there are two functions that are of interest if you want to manipulate these little structs, these kmer count structs. You can ask for the kmer by saying mer, and that will give you like the actual kmer. And you can ask for the count, the frequency, right? So what we're going to do is, so that will just explain this array comprehension I'm about to write. So I'm going to create a variable called filt kmers, open the square brackets for the array comprehension, and say mer of x for x in read k counts. So that will get the kmer from every read in our kmer counter, but here comes the important part. If freq of x is greater or equal to, now from the plot we saw that there was a clear separation between the error and the main distribution between between those two distributions, and so I mean there's a range of values in that trough that we could use as our filter. I'm just going to say 10, because that trough was from like 0 equals like 5 until like x equals like 5 until x equals 20. So I mean anywhere in that range would probably do a pretty good job. So I'm going to say the frequency has to be greater than 10. So this is going to give me the kmers for which their count is greater than 10. Okay, we do that. And again, because it's pretty printing the array of kmers, it has shot off the screen. So let me just bring my prompt back so you can see it. So we have our kmers now that we want to use to build the graph. So let's build the graph. So I'd like to clean up the API and make it a bit neater in the future for people to do this, but right now what you do it is simple enough. Like the first thing you do is create an empty graph, and then you build the graph using those kmers. So say graph, and you say I want an empty graph, and I want the nodes. So the graph we're going to build stores sequences in the nodes, and then overlaps between the sequences in the links. Some different graph formats using different assemblers store it in a different way. The sequences are in the links and not the nodes. It's like, whatever. Like nodes are in genome graphs, nodes are sequences, links are overlaps. And I want the sequences. Here I specify the type of sequence I want to use. And so I'm going to plug in a type that's actually defined in the biosequences package of BioJulia, which GenomeGraphs uses and re-exports and stuff for you, so you don't need to worry about it. But so you can say I want to use long sequence DNA alphabet. Alphabet 4. Let me check that if I spell that correctly. Long sequence DNA alphabet 4. Yeah. So this is a sequence type from biosequences that is optimized for storing longer sequences, and it's going to store DNA as opposed to like RNA or amino acids or whatever else, hence the DNA alphabet, and it's going to use 4 bits to store every nucleotide, so it's compressed. Right? Okay, so that's given us our empty graph that has no nodes, nothing, it's completely empty, nothing interesting. So now we're going to fill it and or construct it. And we're going to do that with the dbg method. So dbg stands for de Bruijn graph. So say dbg exclamation mark because obviously Julia, this being Julia, the convention is name functions with an exclamation mark at the end if that function edits either of the arguments. So it's going to be editing the graph, so I should say mutating any of the arguments, so it will mutate the graph. So I'm going to say graph and filt k-mers. So these are the k-mers that we selected that we wanted to use for construction, and hit enter. And it will tell us it's now constructing a compressed de Bruijn graph from, is that 4 million? Yeah, 4 million odd 31mers. It's constructing the unitigs, and when it's done with that, it will identify the overlaps between unitigs and output the graph. So a few things I should probably explain. Compressed de Bruijn graph. Well, you saw in the demonstration every single k-mer was one node in the de Bruijn graph. That's true, but in reality, in the implementations of a lot of graph builders or graph construction algorithms, what happens is actually rather than construct a whole graph of many nodes, and identifies just from the k-mers input, like where you have these chains of nodes with a single like in and out, like these long simple chains, and just collapses them into one longer node to begin with, and these are called unitigs. So it's like a de Bruijn graph as the nodes are added, like they're nodes that can be like compressed are compressed as they go. So here we go. At the end of that, we got 2,375 nodes, these 2,375 unitigs. It identified the overlaps between them, and it linked them by their overlaps, and it finished. So that's completed the graph. From here, you could do something like, you can say write to GFA1, give the graph, and then like a base file name or something, and then hit enter, and it would write out to a GFA file, which is again a text formatted file that is used in bioinformatics as like a standard to input and output graphs from different softwares. Graphs representing genome assemblies, to be specific, not like graphs that mathematicians are interested in or anything like that. And then you can open that up with like some graph visualization tool. One that we quite like is one called Bandage. I won't go into like using Bandage and stuff because it's a bit beyond. I just wanted to show off like how to do this stuff with Julia today. But what is interesting is a method of assessing the quality of your graph, of your genome. And so remember back to our 3 criteria, the correct motifs, correct number of times, and in the right order. Rather than kind of show you a graph and be like, oh, these are all the nodes, and this is how they link and stuff, and this is how long they are, isn't it great? I'd rather show you another thing you can do to check not the length of pieces of your graph, but the content of your graph. So remember those 3 criteria, the right motifs, the right number of times, in the right order. What we want to see is that when we constructed our de Bruijn graph, or our unitig graph, you'll notice I use the 2 terms interchangeably because they kind of are used interchangeably. What we want to make sure the genuine content from the read data made its way and got incorporated into the graph. We don't want motifs appearing in the graph that don't exist in the data set, because if that happens, that means your algorithm is making up stuff that doesn't exist. That does happen, can happen. Not with this algorithm because it's far too simple, but when you get to more complicated assemblers that try and do heuristics to make decisions about how to untangle some of these difficult regions that I mentioned earlier, then mistakes get made, right? Because it's a heuristic, it's not infallible. But the de Bruijn graph constructor at this stage is so simple and so like standard that it's difficult to make mistakes other than just programming bugs. the logic is sound. The heuristics are sound. So the way we're going to do that is we're going to compare the K-MER spectra that we looked at earlier, that graph from the reads, with a K-MER spectra from the graph. So in order to do that, we need to count the K-MERs in the graph. So I'm going to do that now. We're going to do, I'm going to create a variable, we'll graph K counts SM, so I'm reusing that serial and in-memory K-MER counter that I constructed at the start of this, and I'm going to do a bit of an array comprehension here again. I'd like to implement a neater way of doing this in the API because I'm about to touch a variable that I don't like users touching it. They shouldn't touch this variable because if they do, there's a chance that they would screw up the underlying structure of the graph, and I kind of like to. I know in Julia everything's passed by reference and stuff, but I do also code in C++, and I do think there is something in the notion of which object owns a resource, and I'd like to think that the graph owns its internal variables and that nothing else is going to screw with them. So what I'm going to do is going to say x.seek for x in graph.nodes. So here I'm getting the sequence for every node in the graph. Now what I'd like to do is replace this graph.nodes and stuff with like an iterator that just gives you the sequence of the nodes without you having to go into the internal variables and stuff. But for now, this works. And just hit enter. That was a lot quicker. And you may be unsurprised to see that most of the K-MERs appear once. So that shouldn't be surprising because, again, remember that the Bruijn graph collapses all these motifs, so even the repetitive stuff only should appear once, and to get the genuine genome out, you just traverse that one node many times. Right? So what we're going to do now is, and for E. coli this is really simple, right? So what we're going to do is, with E. coli you expect there's one haplotype in the main chromosome. So what we're going to do is I'm going to construct a spectra again, but unlike last time, this is like a two-dimensional spectra. It's like a projection of two spectras. So say sect, so the intersection, if you like, that's why I'm calling it sect, is equal to spectra. And instead of passing it one set of K-MER counts, I'm going to pass it two. I'm going to pass it the read K counts and the graph K counts. That's that. And this is like a 2D histogram at this point, right? And then we're going to do a custom plot that is in K-MER analysis Makey, and it's a recipe. There are some things I'd like to improve about it, but I'm only just kind of getting a handle on how you make recipes. And so we'll just use what exists right now, and if anybody's got any tips, then I'd be really, really interested. So, please give me a ping and hit me up if you know how to do some of these things. So the plot is called a spectra CN. So C, where CN stands for copy number. So this is a spectra copy number plot. And I'm going to construct it using the sect. Oh, hold on, sorry. I've just realized that you guys will not have just seen that screen, so sorry. Let me do sect again so you can see that. Because I just realized it will go off the screen. Apologies. So yeah, you have your sect. Now we're going to do a spectra cn of that sect, enter. Okay, so that's put the plot. Right. Allow me to switch your view over to the plotting. Let's transition over. There we go. So again, as you can see, quite large x and y axis like last time. So I'm going to have to put the limits on it. So here we go. Limits equal limits. There we go, that's better. So this looks like the histogram we saw earlier, and indeed in many respects it is, because every bar is generated from the KM accounts of the reads of our raw data. But the colours, so the bars and the and you know how high they are, and the x-axis. This is the same. In fact, it's the exact same histogram as we looked at before. It's the KM spectra of the reads, but we've added the KM spectra from the graph, which has added this color overlay on top. Now, this one thing, I'd like this plot to have a color legend somewhere. I haven't worked out how to do it yet. So, if anybody knows how to like add color legends and stuff in plot recipes, send me a message. I know people do it in like Mackie layouts and all this kind of thing, but I kind of just want to do it with vanilla Makey if I can. And I'd also like to have it so as like the x and y axes can be labeled. I can label them after the fact, but I kind of like them to come out labeled from the recipe. But I don't think that's possible right now. So anyway, colors. See the bars from the error distribution are colored in black. So black means any bar is if any portion of the graph is black, then it means these are k-mers that appear no times in the graph. So it's really, really nice to see that all the bars which we wanted to eliminate and did not want to include in the graph are completely black. And all the k-mers we did want to include in the graph are completely, the bars representing that distribution are completely red. So it looks like we have successfully rejected the content we didn't want, kept the content we did want, and there are no other colors. So what you would see if this had gone wrong, let's say there's some content in the main distribution that didn't make its way into the graph because of a bug or something else, what you would see is it's like a stacked bar plot effect, right? So you would have this main red peak would have red, would have some red coloring, but then there would be like a smaller peak contained inside it that was colored black. So it's like, uh-oh, some of this distribution isn't making its way into the graph because part of it has been colored in black. And there are different colors. There isn't just black and red. There's like a blue, which is like if it's in the graph twice and stuff, which is important for when you start wanting to assemble diploids, like, you know, humans and stuff like that. But, you know, black and red is basically like if you want to assemble bacteria, you know. I think like orange as well and stuff, which has been important when we've wanted to assemble triploids and things. So I'd like to maybe do some streams in the future where there's some more complicated scenarios. But for the purposes of like diagnosing whether the construction of the E. coli graph was successful, this shows that it is, because the content we didn't want appears 0 times in the graph because it's black, and all the content we did want to include did make its way into the graph because it's colored in red, and it's, you know, coloured completely black and completely red. So that means the algorithm has done a good job. So that's how you assemble a De Bruijn graph in BioJulia. That's all I really wanted to show people today. In the future, so GenomeGraphs is not going to just be how it contain these graphs. It's going to contain ways of projecting information onto graphs, making some heuristic decision, and then editing the graph. Because actually those 3 steps are essentially what most assemblers that try and do more advanced scaffolding and stuff to make the genome more contiguous, that's all they do, right? You project a genome, you project information onto the genome graph, you look at that information, and then there's a heuristic that tries to use that information to make a decision as to how to resolve a genome and how to edit it to correct some of these pieces of the De Bruijn graph that are hard to resolve, and to make more contiguous. So even mapping reads and stuff, which should be a good one to do fairly early, like how to resolve some simple repeat structures and stuff by just mapping the paired end reads back onto the graph and then connecting up nodes that should be connected. But these are all kind of more advanced techniques, and what I want to show with this framework as I continue developing it, and if I continue doing some of these live streams, is show you how you can do these genome assembly and analysis steps without using like a black box assembler, because in our research in the genome assembly group at my institute, what we find is like most of the advances we've been making in haplotype specific de novo genome assembly is that we'd often find ourselves running like a black box assembly program from a command line and like trying to take a look at the input, look at the output, and then figure out from the output like where it went wrong and all this kind of stuff. But actually, if instead of having like a black box assembly, you open the box and spill all the components out into an API, which is essentially what this GenomeGraphs package is intended to do as it becomes more and more complete, you can run through every, like we've just done now, we've run through every step of building a De Bruijn graph and done like checks every single step. And what I hope is that basically the process goes from being like push, wait, and then be like, oh, it didn't work, to, well, actually the user is much more involved in every step of a genome assembly and diagnoses all the problems. So you put together your genome graph, maybe you run a heuristic that simplifies some of those more complicated regions. You look at the graph and it's like, okay, but well, why didn't it? And then you can like take a step back, look at the information that got projected, be like, does that region have like, is the information that's projected onto the graph in that region really poor so the heuristic can't make a decision? Or like all these kind of things where the entire process is much more transparent and, you know, there's no black box magic about it. So I hope that was interesting for you guys. Yeah, I hope to do some more streams to do with not just genome assembly, but other aspects of BioJulia development, maybe even some streams where instead of just walking through stuff, I might even just, you know, just sit down to code and fix some stuff or implement some extra features in other packages as well, like, you know, that people want, and just sit and talk about it, how the packages are structured, how they work, and some of the design choices that we've made. Okay, thanks very much for watching. Appreciate it. And I will see you some other time. Okay, thanks.